In this video, we are going to talk about the cytoplasmic membrane and the cytoplasm of the bacteria and different components that are present in the cytoplasm. Starting with the cytoplasmic membrane. The cytoplasmic membrane, which is otherwise known as the cell membrane, is present just beneath the cell wall. It is composed of phospholipid bilayer, similar in microscopic appearance to that in eukaryotic cells. So they are chemically similar, but eukaryotic cell membranes also contain sterols, but the prokaryotic cell membrane do not contain sterols. But there is an exception, which is mycoplasma. Mycoplasma is a bacterium that have no cell wall. The only outermost membrane of this bacterium is the cell wall, and it also contains sterols. So there are several important functions of the cell membrane, as it is involved in the active transport of materials from the cell to the outside or from outside into the cell. So active transport is the transport of materials against the concentration gradient, and it utilizes energy in the form of ATP. Then we have the cell membrane involved in the energy generation by oxidative phosphorylation. The cell membrane is the site of synthesis of precursors of the cell wall, and it is involved in the secretion of enzymes and toxins outside the cell. The cytoplasm has two distinct areas when we see the cell of the bacteria under a microscope, an electron microscope. First is the amorphous matrix that contains ribosomes, nutrient granules, metabolites and plasmids. Other one is the inner nucleoid, which contains the DNA of the cell, the chromosomal DNA. Ribosomes are the site of protein synthesis in the bacterium. The prokaryotic ribosomes are smaller in size than the eukaryotic ribosomes. The ribosomes are composed of two subunits. One is the larger subunit, which is of 50S, and the smaller subunit, which is of 30S. Together they make up a complete ribosome of 70S, whereas the eukaryotic ribosome are of 80S, and the larger subunit is of 60S and the smaller subunit is of 40S. The difference in both the ribosomal RNAs and protein constitute the basis of the selective action of several antibiotics that inhibit bacterial protein synthesis but not human protein synthesis. The bacterial cytoplasm also contain several granules, and these granules are the site of storage of different nutrients. These granules are also stain specific, and they can be used for identification of a bacterial species. The nucleoid is the area of the cytoplasm in which DNA of the bacterium, the chromosomal DNA of the bacterium, is located. The DNA of prokaryotes is single circular molecule, and it has a molecular weight of approximately 2 into 10 power 9, and it contains about 2000 genes. The nucleoid contains no nuclear membrane, and it has no nucleolus, and it has no mitotic spindles and no histones. So the major difference between the bacterial DNA and eukaryotic DNA is that bacterial DNA has no introns, but the eukaryotic DNA does have introns.